Professor Chalfie, thank you for accepting our interview request. You are one of the world's most respected scientists, but your interviews and personal charisma suggest that you are much closer to the young man who had just earned his PhD than to an unapproachable researcher. How did winning the Nobel Prize affect you?
So the Nobel Prize affects people, I think, in different ways. I like to joke that someone said to me after getting the Nobel, well, now you have power. It didn't really have power, but now you have power. What are you going to do with it? And then I usually add, that was my wife that said that. But I think she was making a very important point. If the Nobel gives you visibility, I think there's an obligation to use that visibility. So what I've done is what I believe many Nobel laureates have done, and that is we've been teachers almost all of our lives. We're going to go out and talk more with students. If that visibility means that we can reach more people, that's wonderful. But there's been lots of other actually wonderful opportunities, because the visibility has meant that I've been invited to come to countries like Hungary that I had never been to before, and probably would never have been invited had it not been because of the Nobel. There's not a problem with that, but it's sort of a truth that I've visited many more places in the world since getting the Nobel than I ever did before. And that's a wonderful opportunity to meet people, to talk with people, to find out how they feel about things. It's wonderful. I've also been privileged to be asked to be part of different organizations that I think if I had not had the Nobel, I don't know if I would have been asked. So in 2014, or before 2014, I was asked if I would run to be president of the Society for Developmental Biology in the United States, and I was their president for that year. And more recently, I was also asked to run to be president of the American Society of Cell Biology. So these are wonderful opportunities to give back to one's community, and so I'm sort of honored that they would consider me and that it would give me a chance to give back something in that regard. So all of these things are really very good. I think there is a problem, though. Yeah. And our problem is we sort of honor the Nobel and Nobel scientists maybe a little too much. It's not the best scientist in the world. It's not the smartest person in the world. It's not the one with the most students or the most funding or anything. I think the Nobel goes to people whose work had a profound impact on the way we think or do science. And often you cannot predict whether your work is going to do it. I don't think that's something you work for. And so many times it's, I think we're all very concerned about our work. We're all very passionate about the work we've done in our lifetimes. But whether someone is a Nobel laureate or is not a Nobel laureate is sometimes not in anyone's hands, because it has to do with what has been the impact of the work. Have people used it? GFP, to give one example, people by the thousands used it in experiments. It had a very big impact. I knew it would be useful. I thought people would like to use it, but I had no concept of what it was going to do and what it was going to be. And so I could never have predicted that it would have had that impact.
You were interested in the topic.
I heard about the protein. I thought it would be a very useful tool, and it was more, this is going to be fun, let's try it and see what it is. And if it works, it's going to be very useful. But how useful, what people were going to do with it, and what the consequences were going to be. So I'll give you one consequence. GFP is a protein that normally you shine blue light on it, you get green. But you can modify the protein, as Roger Chen did, to make it one that you shine ultraviolet light on and get blue, or you shine blue and you get a yellow-green. But you can't you can't get any other colors beyond yellow-green. You can't get a real yellow or an orange or a red. And for several years, no one had any idea about what to look for, or how to get something like that. And then Lukyanov and Matz in Moscow and their co-workers showed that there was a red fluorescent protein in coral. Not only did they give us a new color, but they also said basically to the world, there's a wealth of interesting molecules out there that no one has paid attention to. And it sort of spawned a sort of little mini industry of finding other fluorescent proteins. I think the number now is in excess of 1,000, probably 1,050. Last time I checked, it was 1,024. So just an enormous number of proteins. So I would never have predicted that people would have gone off to find strange or unusual fluorescent proteins or anything. So that's what I mean. You can't predict what happens. You can't predict the impact. But in general, it has been a great deal of fun and a really wonderful opportunity. Now, what doesn't happen is, we scientists in general do put this prize on a pedestal. And we think, if that's the best you can do, everything wonderful must happen to those people. Because if it doesn't happen to them, it's not going to happen to me. It's like, and so I think there's a little bit of, I'm going to exaggerate it, but there's a little bit of an imagination that there is a long line of people outside my door with bags of money saying, please let me fund your research. Doesn't happen. And the journals don't write me and say, please just send us your lab notebooks. We'll write the papers for you and it'll be published. That doesn't happen either. You're a working scientist. You're like everybody else. And that's what happens.
Did the rate of rejection of your papers change after winning the Nobel Prize?
No, it wasn't that much different, but they were rejected. I have had papers that, the reviewers still come back and I wonder why they say the things they do. But many Nobel laureates that I know, and I was one of them, have had grants turned down after the Nobel. And that's good, because you shouldn't get some sort of free pass because of some work you happened to do 14 years before the prize. You should be judged on what you are going to do and what the work, what evidence you have for your work. So we shouldn't get a free pass, and we don't.
I read some interview when I was preparing for this one, and I learned that you were interested in many things as a child, but you ended up in life sciences. What influenced you when you started your university studies in this field? So when my brother, one of my brothers reminded me several years ago about an event that happened in high school. I had gotten good grades in high school, and there was a big pharmaceutical company that was in my town, and they gave a big scholarship to the science student from our high school. And I went to be interviewed, and the interviewer said to me, so what is going to be your major in college? And I said, I'm not really sure. I don't know yet. I'm going to wait until I get to college to find out. And he said, but it is going to be in a science, isn't it? And I said, I'm not really sure. And he said, you're not eligible. He sent me home. And according to my brother, my mother said, sometimes you shouldn't be so honest because I didn't get a chance for the fellowship. But
When I went to college, the first course I took was a second-year calculus course. And the teacher was wonderful, stimulating, encouraging, just a wonderful person. I actually went and saw him about 5 years ago, and we renewed the meeting. And so for a short time, about a year and a half, I thought, oh, maybe I should be a math major. I like math. I took quite a number of math courses, but ultimately decided that maybe I really didn't know what I was doing, and math may not be the thing for me. And I was sort of trying to figure out what I should do next. And I wasn't that interested in majoring. I had taken chemistry courses and physics courses, but I wasn't that interested in majoring in chemistry or majoring in physics. I had taken I think I'd taken one biology course. Mm-hmm. But there was this major. It was called biochemistry, and it just sounded sexier. It just sounded much cooler.
Biology plus chemistry.
Yes, so biochemistry. And the way they had set up the major, I could use all my math courses to satisfy the requirements. And plus I had to take, I did take other biology courses and so on. So it was more, it just sounded new and different and exciting. And so I ultimately majored in biochemistry.
And after graduation, your parents encouraged you to pursue academic career. Apart from this support, what else motivated you in this direction, so to become a scientist?
So, my parents were always supportive of me, but I don't think they encouraged me to be an academic. So I don't think that's quite true. But what happened when I was in college, because I thought I was going to go into science, I worked the summer after my second year and the summer after my third year in two different labs. One was in the Chicago area where I lived, and one was at Harvard where I was an undergraduate. And that experience after my third year in college, after my junior year, was horrible. And I was the cause of what made it so bad. Because I had it in my mind that real scientists, whoever real scientists are, were people that just knew what to do. They knew how to do it. And if you didn't know how to do it, that was proof that you weren't really a scientist. I don't know how I got this silly idea, but it was definitely in my mind. I also had joined the lab somewhat later than when everything was, and so I wasn't with anybody. I was all alone in a room 50 meters away from where the main lab was. So there was no one for me to talk to, no one for me to ask, am I doing the experiment right? And all the experiments failed.
Oh, not too motivating.
So throughout the entire summer, everything failed. At the end of the summer, the head of the lab said to me, you know, we have a couple of weeks before the term starts again. Why don't you go home, rest, relax, come back and try it one more time. If it doesn't work, we're going to have to stop the project, but try it one more time. And I did that. And it failed completely. And I said, that's it. I have given myself proof that I should not ever be a scientist. And with that, I finished my last year of college taking the one remaining science course I needed to take and courses in all sorts of wonderful subjects that I really enjoyed and had a wonderful time. But I graduated really not knowing what I was going to do. And so I went and did a series of jobs. Peter had mentioned this in his introduction. I worked immediately after I went home, and I worked as a janitor in the elementary school near where I lived for a short time, and decided that was too hard of work to do that. Then I had worked in a factory. After my first year in college, I went back to that factory and worked there for a couple of months, and that wasn't any fun either. And had a couple of, I took some courses, and after I took the courses, I had to find a job. I happened to find a job interviewing people in hospitals at various cities in the United States, but it only lasted 1 month, and then they said, thank you, and you've done your job. And at that point, my father had unfortunately been found to have colon cancer. And my father was the sales part of the family business, which was dress manufacturing. And so he couldn't take the line, the dresses, to the stores on the eastern part of the United States because he was recovering from surgery, which he recovered completely from, which was wonderful. But I didn't have a job then, so I took over his job. And for 1 month, I went to all these different dress shops, not knowing anything about the business or anything at all, and would walk in, and they so respected my father that all I had to do was open up the bag and let them see the samples, and they would say, oh yeah, but this one will go to this one, fine. And they picked out what it was. And so it was the easiest job in the world because of the respect all these store owners had for my father, and the particular way he was of dealing with them, which was a wonderful thing to learn.
But what did you bring back to the lab after this period?
So after all of that, I got a job teaching high school. And when I taught high school, I made a very important discovery. High school students have the summer off. They have summer vacation. High school teachers have to find a job. So I needed to find a job. And so I went to, I had a friend at the school I was teaching at, and she said, I think I know someone who you could work in his laboratory. I said, well, okay. And I went to work in the guy's lab. And in the interview, he was telling me about his work, and it made me remember a paper I had written for a college course. And I remembered the conclusion of the paper, so I asked him, does this happen here? And I think he was so surprised I asked him a question that he hired me. I had really no skills, but he hired me for the summer. And so I had thought of an idea, I had a question. Then he assigned me something that was a horrible experiment, and then he left for the whole summer. But there were other people I could talk to, and particularly I found someone who had also asked him the same question. Does this work in this particular way? And so we were talking, and I said, how would you test that? And he told me, and I was able to learn the procedure, and I tested it, and it worked overwhelmingly well. It just exploded. It was wonderful. And I could contribute not only the initial experiments, but I could go to the library and find the relevant articles that backed up the idea. And so when the boss returns at the end of the summer, he asked me about the experiment he had told me to do, and I said, I'm sorry, I didn't do that, but here's all the things that I had done. And he was very supportive and very happy. That became my first publication. Mm-hmm. And with it, he suggested that I go to graduate school. And so that was really the motivation, to having that little bit of success. Yeah. And that little bit of support was what gave me the confidence to apply to graduate school. I should also say that there was a somewhat negative reason of why I went to graduate school. This is going to sound a little arrogant, and I don't mean it to be that, but as a young 22-year-old teacher at the school, I had no problems with the students, but the administration basically ignored, it would not count my opinions as being important. And I didn't like not being considered an equal partner or someone to be listened to. I remember one faculty meeting where a question came up and I said, you know, I think logically we should do this, this, and this. And the head of the school looked at me and said, Marty, that is very logical, but I'm just telling you, we're never going to do it. And that was it. I mean, you know, we're going to ignore you. And I didn't like not getting the respect that I thought I should have, that I should have been an equal partner in all of this. And instead, I was just discounted. And I thought, well, maybe if I go to graduate school, I will be treated a little bit better.
Maybe the world is lucky that you did not feel yourself well in that school. Maybe this way.
I don't know.
Maybe you wouldn't get the Nobel Prize in that high school. When you started working on a green fluorescent protein, what made you start working on this specific research topic? What was the starting?
So the initial start, or before the actual start, was the type of work I was doing, and have continued most of my scientific career. I used genetics to understand how nerve cells develop and how they work. And in particular, I had been working on a series of mutants of the nematode, this roundworm, Cinerabdias elegans, and these were mutants that were defective in touch. Some of the mutants caused the cells never to be made, and so those mutants allowed us to start studying what are the controls that make particular types of cells. Other mutants were defective in touch because the cells were there, but they didn't work. And so those started leading us to find the molecular basis for the sense of touch, both of which were very exciting problems to me. And the one thing you should know about this animal is that it's transparent. So I work on a small 1 millimeter long worm that's transparent. And I'm studying a whole bunch of genes that were beginning in the 1980s to clone. Now, when you clone a gene, one of the very first things you want to ask is, because a gene is not turned on, not activated in every cell, you want to know what cells use that gene. What cells turn that gene on? And there are a lot of methods to do that, but they're cumbersome. You have to basically kill the animal, fix it, and permeabilize it, and treat it with chemicals, and do a lot of work with it. But we were able to answer the question. We knew that gene A was in these 3 cells, and gene B was in whatever. But that was what we were working on. We were cloning genes in a transparent animal and wanted to know which cells use those genes. And I go to a seminar in 1989, and I hear that there is a protein that is fluorescent. That the only thing you need to see that is shine blue light on it, and you will see it as green. I work on a transparent animal. If I can get the gene to make this thing, it'll be made in the right cell. I shine blue light through my transparent animal, and whatever cell is green, that's the answer. But not only that, I don't have to fix the animals. They're alive. I can see if the cell changes shape, if it grows, if it goes to a different position, and I can start studying all sorts of questions. So I'm listening to the seminar, and I'm basically getting more and more excited, because I want to try this tool in the cell.
Right time and the right place.
And so, yes, exactly. So the next day, I find out that there is a person named Douglas Prasher, who is in the process of isolating the DNA for this gene. And I call him up and we have this wonderful conversation for about an hour about how we're going to take that gene the minute he gets it, and we're going to put it in the worms, and we're going to see the cells if it works. So we both got very excited about the idea, but that's what happened. That was the initial excitement. I was so excited when I heard about the protein at the beginning of this seminar that I don't know what the seminar was about. I just kept thinking about what were all the exciting things I wanted to do with this? What were other people going to do with it? It was going to be a wonderful tool if it worked. I should say that I learned very quickly that nobody believed it was going to work. And there was a very specific reason for that. Proteins are made of a long chain of amino acids. And the atoms that connect it is called the peptide backbone. And it's just a line of atoms. GFP starts off like that, but then does something astonishing. 5 of the atoms make a circle. So instead of having a line, you have a line and a circle and another line. And no one had any idea how that circle was made. And if you don't know how the circle, it was known the circle had to be made or it wouldn't work. So no one knew how it worked. And so everyone imagined that there was going to be something that would be called a converting enzyme, converting it from the line to the line with the circle. Or it could take 2, or maybe 5. No one knew. But what they were pretty sure of is it needed something. And if it needed something, it wasn't going to be very useful. Because then you'd have to put the GFP and whatever else was needed. And especially if it was 2 or 3 or 4 things, that would make it much too complicated. It wouldn't be a useful tool. So they all said, it's not going to work. The few people that knew about it. We just tried it. We just tried to see if it worked. It turns out that the molecule is its own enzyme. So the molecule causes itself to reorganize itself like that. So it does not need anything else. And that's what made it a very useful tool, because you just add it, put the DNA in an organism. Once the DNA is in, that DNA goes to the progeny, and their progeny, and their progeny. You've made a line that has this thing in it. Then whenever you want to see it, you just shine the blue light on it. And you could look at it in living animals, living cells. It turned out to be very, very powerful.
Very fascinating story. I think you remained an active researcher after receiving the Nobel Prize. After such a big recognition, what motivates you to keep working?
So I like to tell people that the work we did with GFP was published in 1994. And the Nobel Prize was given in 2008, 14 years later. What was I doing in those 14 years between those things? I was working on these genes that were needed for cell development and for touch. And we were working on them before GFP, after GFP, and after the Nobel. So we've had, there's been a lot of questions that we've tried to address, and we look at it, we're still looking at things. It's fun to do the work. It's not that you get the recognition and then you say, okay, now I can retire. It was a wonderful marking point to say, this is the recognition of the impact of this molecule. I actually believe that the three of us got the prize. It wasn't to the three, the prize was to the molecule. The molecule was useful. But but I was doing stuff before that. I didn't want to suddenly say, okay, I'm done now. Still a lot of questions I want to address. And I think quite a number of people that have Nobels want to continue working if they can. Because that's what brings us happiness in this. And that's, I think, an important thing.
Maybe the best motivation. Yeah. Currently, you are participating in the conference of the National Academy of Scientists Education in Hungary, Szeged. Do you think that supporting talented young people can help really them to have a better chance of developing their talents? Or would it be better to leave them alone and try to find their own ways?
I think any chance to give an opportunity to students is well worth it. So I don't think, I mean, if there was a way of giving opportunities, but giving them some freedom to do it, that would be good. But I think these are the people that are going to be the scientists of the future. And if we don't encourage people to look at things and take the same wonder I think that we do in the world around us, I think that's sad. I don't want everyone to be a scientist. I do want people to sort of understand the sort of ways we think about information, what is evidence, what is risk, various things, what statistical significance. I would like more people to understand that. But in general, I think we should be giving as many opportunities for students to really grow into being scientists. Now, that said, I want to say something slightly different. I think we should also do that for musicians and artists and actors and people in the arts. I think that there sometimes gets to be a sort of almost utilitarian argument that, well, we need scientists. That's good for the economy. We should push science. I think we need to push the humanities, the arts, the social science. I think we need to do everything and give opportunities. This is a wonderful program. I'm not saying it's not, but I don't think it should be the only program.
And have you seen similar programs somewhere in the world which is working similarly than this one? Not exactly like this, but there are efforts. There's lots of science fair programs that are sponsored in various countries that give students chance in high school, for example, to compete with projects that they come up on their own. I think those are wonderful. Anything that gives them an opportunity. I think there's, I was very fortunate to be asked to be part of the board of an organization that's now called the Society for Science. They put on several scientific competitions for high school students and now even younger students, and that's good, but they also put out a wonderful general science magazine called Science News. And Science News is now being sent to a very, I'm going to say 40%, but I'm not sure, of all high schools in the United States. Being able to reach people, to have them find some enjoyment, some awe in looking at science, I think is really important. It's not this thing, but there are other efforts. I'm not as familiar with them, but I very much like this effort. I like the fact that students get a chance to be excited about what I would say more modern science, more molecular biology, more thinking about the genetics, the things that most of us
in the lab actually do think about. And I think getting that taste early is a very good thing.
I think role models can have a major role in career choice. That's why it is important to invite famous and motivating scientists like you to give lectures here. Beyond personal examples, how do you think biomedical science can be made more attractive to young people?
So, I know of several different efforts that people have made at various times, and most of these are at the college level, where it's basically involvement more than anything else. And I think this program is that at the high school level. It's involvement with doing it. It's not reading about it. I've got my hands on the machine and I've seen how this happens. I see what goes wrong. I see what goes right. I see how I'm thinking about this, what I should be thinking about, what the problems are. And so I think there are a number of outreach programs. I think there should be more. I think they should be done younger. I'm going to say a little strange thing here. In the United States, my belief is that if you ask any child, 11, 12, and younger, what is your favorite museum in New York City? They all answer the same thing. It's the American Museum of Natural History. If you start asking them when we're 13, 14, and 15, do you have a favorite museum? If they have a favorite museum, it's never that. It's the Museum of Fine Arts or the Museum of Modern Art. It has changed because somehow, and I don't understand this, science, going to the Natural History Museum, going to the planetarium, that's something that kids do. That's not, as I get, if I be, teenager growing up, I have to do more adult things. No, that's really not the case. But somehow there's a little bit of a hint, maybe because kids, when they're 9 years old, they're in 4th grade, they've learned how to add, multiply, divide, subtract. They know about basic math. They know how to read. The world is open to them, and they start getting excited about dinosaurs and birds or whatever it is, and they're very excited. That's something. Somehow, it's the excitement is there. They're excited about the stars. They're excited about the planets. They're excited about all sorts of things in nature. But somehow we're losing them. So it's not that the interest isn't there. The interest starts. And I'm not entirely sure what we need to do to maintain the interest. I think the program here is doing a pretty good job of it, though.
Yeah, today we are lucky because we had an audience full with young people who are interested in science. What impressions did you get when you met the students from this academy today?
So I liked them very much. I liked their questions. I liked talking with them. I thought they were certainly much more sophisticated than I was when I was their age in school. They're thinking at a much more, I don't want to say sophisticated, but at a much more involved level, and seeing something that's really interesting about the world around them. And I think that realization is a great start.
Coming back to the start of this interview, what is your personal strategy to stay motivated if your experiments fail or the results are not at all what you expected? Because you had such periods in your life.
We all do. This happens to everybody, that the experiments don't work or they change. I've said this many times. I work on mutants that affect a cell either in its production or its function. Sometimes the studies on one aspect are going quite well. and the other ones are not. So as long as one is working, I'm perfectly happy that the other one will sit for a little while. So doing multiple things, I think actually helps. Having multiple things going on, that maybe one person in a lab, their experiments might not be working, but another person's experiments are working. So it's a hard thing to tell somebody, you should be doing more things and that'll be the way to solve this. But I think having multiple problems that you're thinking about does help this. I'm a fairly enthusiastic person. I think that's one of the main attributes I bring to things. I get excited. I really get excited about ideas. And I think I convey that to the people in my lab, maybe annoyingly so, but I'm pretty sure that I do it. And I sort of enjoy the results. I like hearing about science. I like hearing about new things. I like hearing how things work. I like it when people have done clever experiments to figure something out. It's just exciting to me. So I don't feel I have to maintain that. That's there. And there's so much around us to be stimulating that maybe I've gotten to a point in my life that there's a backlog of things that I'm concerned with or wondering about, and so there's a lot of things going on. But I feel that there's a lot of excitement out there. As I said to the students earlier today, we are right now in the golden age of science. We're not, you might talk about the golden age of movies or opera or art of one type or another, or theater, but the golden age of science is always right now. It's always going to be right now because we keep learning more things and we have new approaches and new excitements. And if I look back on my academic career, and what we could do when I started my postdoc in 1977, or what we could do when I started my faculty position in '82, or when did cloning really take off, which was sort of the late, sequencing DNA started in 1977. And it just took off from there. And then you get in 2003 or 4, finally, the really good draft of the Human Genome Project. So that's an astonishingly short time. The discovery of RNA interference, the discovery of genes having to do with development, the discovery of genes having to do with developmental timing, the discovery of cell death genes, the discovery of microRNAs, all the CRISPR-Cas9, cryo-EM, all of these things have come, and each one of them is a spectacular addition. And there's going to be many more. So it's always, there's a point of excitement.
Yeah, maybe it's not a better motivation than that we have more and better tools than ever, and more and maybe most important questions than ever. And this
I think so, but I also think we still have some of the old questions. Yeah. We really want to know how all the parts of the cell interact together. We know the parts, or we think we know the parts, but how do they work together? How do they interact? Next year is the 100th anniversary of a very famous cell biology book in the United States. It was by E.B. Wilson, one of the giants of early cell biology. He happened to be the chair of the zoology department at Columbia, where I am. And in 1925, he wrote the third edition of his classic book. It's over 1,000 pages, summarizing all of cell biology. And he gets to an interesting question at one point. He's talking about the cell. And he says, you know, the cell is the unit of life. It's living. But the nucleus isn't living, and the ribosomes, well, he doesn't call them ribosomes, but the nucleus and the secretory thing, the parts are not living. How do we get a living cell? We still have that question.
Yeah.
How does this fit all together? How does this work together?
Where is
So
the border?
we have the old questions still to answer. They're very fundamental. And so I think that's an important thing to think about, that we have but we also have new questions that we never imagined before, and new approaches, and new ways of thinking, and new ways of observing that just were not there before, and they allow us to do so many new things. Tomorrow, I'm leaving for Iași in Romania for the 25th, for celebrating the 25th anniversary of George Palati's Nobel Prize. And what George Palati did was use the electron, he was one of the first people to use the electron microscope to really look at what the parts of the cell were that Wilson had no concept about because he couldn't see with that resolution. The electron microscope changed our whole image of life. And George Palati was the one that introduced so much of that. So it's all the technology coupled with people's enthusiasm and thoughts and concepts and ideas, all of that sort of makes for a very exciting time.
Thank you for the thoughts. Thank you for the interview. Thank you for your excellent lecture today. Thank you for coming to Szeged.
It's been a real pleasure. Great talking with you.